everyone and welcome back to our channel. So from today onwards we shall begin the drugs acting on the cardiovascular system. That is a new chapter. So before going into the actual drugs which act on the cardiovascular system, it is very important to understand the role of angiotensin. Okay, so we shall deal with the angiotensin in today's video, like how it is generated and what are its different roles and what could be done, what are the pharmacological approaches which are related to this angiotensin. Okay, so coming to the outline of this particular video, we shall first discuss about the Renin-Angiotensin system, which is known as RAS, the accepted short form, or sometimes it is also referred to as Renin-Angiotensin-Aldosterone System, RAA system. Okay, so this is another name because aldosterone is also involved in this particular system. Okay, so this is the first part which we are going to deal with. Next we shall deal with the pharmacological actions of angiotensin. What are the different effects it is producing in our body? So what are the pharmacological actions? Then comes the angiotensin receptors through which they mediate the actions. Next we shall deal with the pathophysiological roles of angiotensin, how it is important in certain physiological processes, or the pathological roles. What are the pathological roles? All these we are going to deal. And lastly we shall deal with the inhibition of this renin-angiotensin system. Okay, so we'll understand why we need to inhibit this particular system from the actions and the pathophysiological roles into which this angiotensin is involved. Okay, so this renin-angiotensin system is the one which results in production of this angiotensin. So let us come to the details of this renin-angiotensin system or renin-angiotensin-aldosterone system. Okay, so now let us consider what exactly this angiotensin is and what is the historical background behind the development of this particular substance which is obtained in our body. Okay, so angiotensin 2. Okay, now as you can see it is numbered, so that means it will be more than one type of angiotensins which is found in the body. Okay, so it is mainly the angiotensin 2 which is an octapeptide. That means there are 8 peptide bonds. Okay, so it's an octapeptide which is basically generated from a precursor plasma alpha 2 globulin. So this is the source. Okay, alpha 2 globulin, this is a plasma protein, it's a type of plasma protein, right? So this substance angiotensin 2, it is generated from the plasma proteins. Okay, and it is involved in a number of functions, normal functions like electrolyte homeostasis. Okay, that is the maintenance of the levels of the electrolytes, that is the ionic composition of the body within the normal range. Also it is involved in the blood volume homeostasis to maintain the blood volume in normal range. Apart from that, even the maintenance of the blood pressure homeostasis. Okay, so your blood pressure is also maintained within a particular normal limits, right? So all these homeostasis, it is taken care of by this particular substance angiotensin 2. Okay, so it has a lot of physiological roles in our body. Okay, and it is obtained from the plasma proteins. Now initially in 19th centuries, kidney extracts were obtained and that was found to be having a pressor action. Now, what do you mean by pressor action? Yes, the one which can increase the blood pressure. Okay, so these kidney extracts were found to possess some kind of substance which resulted in increase in the blood pressure. Okay, so this active principle which was obtained from this kidney extracts, it was named as renin, and later on in 1940s it was found to be an enzyme. So it was studied in detail and it was discovered to be an enzyme. Now, what is the role of enzyme in our body? Yes, they catalyze certain reactions, right, in our body. Okay, so obviously this is not the actual substance which resulted in this increase in the BP. So this enzyme resulted in production of a pressure principle from the plasma protein. So this is what was discovered, that this enzyme which is obtained from the kidney or which is produced by the kidney, it is responsible for production of some other substance which actually is responsible for this increase in the BP. Okay, so this is directly not responsible, but indirectly it resulted in production of one more substance that is actually responsible for the rise in BP. Now this renin, it was later on understood that renin catalyzed a reaction to obtain a decapeptide, and that was named as angiotensin 1. Okay, now decapeptide means there are 10 peptide bonds, right? So angiotensin 1, it was found that later on it gets converted into the active principle, that is the octapeptide angiotensin 2. This ANG, this is an accepted short form for angiotensin. Okay, so this enzyme renin, it catalyzes a reaction to convert angiotensinogen to angiotensin 1. So this was a principle which was obtained by the action of renin. But actually angiotensin 1 was not responsible for the rise in BP. But further this angiotensin 1, it gets converted to the octapeptide angiotensin 2. And this is actually responsible. This angiotensin 2 is actually responsible for the rise in BP. Now, how this angiotensin 2 is generated from angiotensin 1? That's under the influence of another enzyme, that is angiotensin converting enzyme. ACE is the accepted short form for this. Okay, so with this brief introduction, we shall see what this renin-angiotensin system is in detail. Okay, so this is the basis for the development of this angiotensin 2, and it was later on understood to be having a number of pathophysiological roles, and the major concern was the development of the hypertension. So that's how the studies led to the introduction of many important drugs like angiotensin converting enzyme inhibitors, ARVs, that is angiotensin receptor blockers, which resulted in the inhibition of the actions of this angiotensin 2. So understanding what is the role of this angiotensin 2 is very important to understand the pharmacology of drugs which are influencing your cardiovascular system. So it's not only in the hypertension, but it also plays in the pathophysiology of other conditions as well. So it plays an active role in the development of congestive heart failure as well, myocardial infarction complications as well. So it has a very wide application. The inhibition of this particular system or the inhibition of the actions of this angiotensin 2 has a very important implications in the cardiovascular complications or cardiovascular pathologies. Okay, so let us understand this particular system in detail. Now, involvement of the system in a lot of cardiovascular condition, it has led to a considerable attention towards this system in the recent years. Okay, so this system has been studied in detail and it is found to be operating at two levels. The one system is known as circulating renin-angiotensin system. Now, what do you mean by this term circulating? So, one system is known as circulating system, and the second system which is operating, it is known as a tissue or the local renin-angiotensin system. Now, what do you understand by these two terms? Okay. Actually, the system and all the steps involved are the same. Only thing is the location is changing over here. Where exactly this system is operating, that is changing. And accordingly, it is of two types, or it operates at two levels. Okay, so first one is known as a circulating renin-angiotensin system. Now, obviously, where it will be found, what is circulating in our body? Both the systems are present in the body. Yes. What is circulating in the body, throughout our body? Yes, it is the blood. Right. So circulating renin-angiotensin system is the one which is operating within your bloodstream. That is why it is known as circulating renin-angiotensin system. So within the blood, all these components are getting generated. We will see what are the steps involved. Okay, and obviously the second one, where it will be operating, as the name implies, tissue or local. That means what? This particular system is operating within certain tissues, within certain organs. Tissues are parts of organs, right? And that's why it is known as local. Local means what? Restricted to a particular region or a particular area. That is the reason why it is known as local renin-angiotensin system, and this one is known as the circulating renin-angiotensin system. So basic difference is what? Where exactly it is operating, or where exactly these steps are taking place. That is the only difference. Okay, now coming to the first one, that is the circulating renin-angiotensin system. So obviously, as I have told you, this is the one which is occurring in your bloodstream, okay, or in the plasma, right? Now this is the diagrammatic representation of this renin-angiotensin system. Okay, so as I have told you that renin, which was found to be present in the kidney extracts, okay, JG cells. What are these type of cells and where are these found? Yes, these are the juxtaglomerular cells. Where they are located? Yes, obviously these are located in the kidney, right? Remember the juxtaglomerular apparatus, which comprises of these juxtaglomerular cells and the macula densa, right? So this particular juxtaglomerular apparatus, it takes care of the blood which is flowing through your kidneys, right? The blood pressure, it's continuously monitoring the blood pressure, right? So these juxtaglomerular cells, which are a part of juxtaglomerular apparatus, it produces an enzyme called as renin. Remember the extract which was obtained from the kidney, it actually is generated by these juxtaglomerular cells. So renin is nothing but the enzyme, right? So the enzyme which catalyzes the first step in the renin-angiotensin system, that is conversion of angiotensinogen to angiotensin 1. Okay, so angiotensinogen, it is a plasma alpha 2 globulin which is naturally found in our blood. So this is nothing but it's a polypeptide, right? So certain amino acids will be clipped off, and this angiotensinogen, it gets converted to angiotensin 1, which is a decapeptide. Decapeptide means there are 10 peptide bonds, right? So 1 to 10. So it tells you which sequence of the amino acids are present over here out of this angiotensinogen. So the amino acids present from position 1 to 10 are retained over here. Okay, so that is angiotensin 1, which is generated by the action of renin on the angiotensinogen, which is a plasma protein. Now this renin, it has a very short T half-life. Okay, so its plasma T half-life is just 15 minutes. So the renin, which is produced by the kidney, it is released into the blood, and normally the blood, it contains this angiotensinogen, so it gets converted into angiotensin 1 in presence of this circulating renin. Okay. Now this angiotensin 1 which is generated under the influence of renin, it is further acted upon by the second enzyme, that is ACE, right? That is angiotensin converting enzyme. Okay, this is also known as kininase 2. Okay, so this is actually a dipeptidyl carboxypeptidase, which is usually found on the surface of the vascular endothelium. What is endothelium? That is the innermost layer of your blood vessels, right? The part which is in contact with your blood. So the surface of this endothelium, vascular means the blood vessels, right? So this surface of the endothelium or the luminal surface, it contains this particular enzyme, converting enzyme or ACE. Okay? So this enzyme converts this decapeptide into an octapeptide. Okay, that means what? 2 of the amino acids are clipped off. Now here it was from position 1 to 10, right? Now here you can see it's just from position 1 to 8, right? So only 8 amino acids. So rest of the 2 have been clipped off. So it's an octapeptide, correct? Okay. So this is the second step. Now again this angiotensin 2 which is generated. Remember this is actually an active principle, right? As you can see, what are the effects which are produced by this? This is not the one which is responsible for any action. It's the angiotensin 2 which is the active principle responsible for vasoconstriction or retention of the sodium and water in the kidneys. Even it secretes the aldosterone from the adrenal cortex. This is the reason why it is known as Renin-Angiotensin-Aldosterone System. In turn, this will promote the actions of angiotensin as well. Okay, so it is also involved in the homeostasis of the blood pressure, blood volume, and your electrolyte maintenance, right? So this is the role of the renin-angiotensin system. Okay. Now further, this angiotensin 2, this also has a very short T half-life. As you can see, it has short. T half-life is just 1 minute, so it's very short, right? So this angiotensin 2, it will further get converted into angiotensin 3. Now this is a heptapeptide, means there are only 7. As you can see, the sequence is from 2 to 8. Here it was 1 to 8, right? So which amino acid has been clipped off? The one which is present at position 1 has been clipped off, right? So that is why it is written here 2 to 8. So amino acids from position 2 to 8 of this angiotensinogen are retained, and that is known as angiotensin 3. As you can see, to a certain extent, this angiotensin 3 is also active, right? This also is responsible for release of the aldosterone, correct? So certain amount of action is also produced by angiotensin 3, but vasoconstriction and retention of sodium and water, that is produced only by angiotensin 2. Hence this is actually the important component of the renin-angiotensin system. Now this angiotensin 3 which is generated, this is further acted upon by the peptidases. So a number of peptidases will act upon the angiotensin 3, and these are collectively known as angiotensinases. Okay, so these are responsible for the hydrolysis or the further cleavage of the angiotensin 3. Okay, so these will inactivate the angiotensin 3. It will get converted to hexapeptide, that is, it has the amino acids from position 3 to 8, as you can see. So there are only 6 amino acids over here, and this is termed as angiotensin 4. Further, it is acted upon by angiotensinases and it gets converted to inactive fragments. Okay, so this is the complete renin-angiotensin system. Now, as we were discussing the circulating renin-angiotensin system, which is operating in your blood circulation, right? Now, coming to the second type of renin-angiotensin system, that is the tissue or local renin-angiotensin system. Now, as I've told you, all the components are same, all the reactions are same, only thing is the location is different. Now, depending upon its location, further this tissue or local renin-angiotensin system is divided into 2 subtypes. That is the extrinsic local renin-angiotensin system and the intrinsic local renin-angiotensin system. Okay, extrinsic, intrinsic meaning something within, right? Extrinsic means something which is exterior, towards the exterior, right? So this extrinsic local renin-angiotensin, now this is not a circulating one, it is restricted to a particular place. Now which is that place? It is on the surface of your blood vessels. Okay, so this blood vessels capture the circulating renin, the renin which is circulating in your blood, that is captured by the surface of your blood vessels. Also this angiotensinogen, which is alpha-2 globulin. So this is captured onto the surface of your blood vessels, and locally on the surface of the blood vessels, angiotensin 2 is generated. Okay, so either within the endothelial cells or on the surface of their walls. So that's why this is known as extrinsic local renin-angiotensin system. It is not taking place within a particular tissue or an organ. That is why it is termed as extrinsic local renin-angiotensin system. Whereas the second one, as the name implies, this is operating within the tissues, inside the tissues, especially the tissues which are found in the heart or brain, kidneys, adrenal. So all these organs, they possess all the components of your renin-angiotensin system, all the enzymes and the precursors required. So angiotensin 2 is also generated within these cells of these organs. Steps are same. Angiotensin-nogen, it gets converted to angiotensin 1 and angiotensin 1 gets converted to angiotensin 2. Under the influence of renin, which catalyzes the first reaction, that is angiotensin-nogen to angiotensin 1, and angiotensin converting enzyme, it converts angiotensin 1 to angiotensin 2, which is the active principle. Okay, so difference is where exactly it is located, and depending upon that there are two types, that is circulating and tissue or local renin-angiotensin system. Okay, now with this brief introduction, let us discuss the pharmacological actions of angiotensin 2, which is an active principle. So let us see what are the effects which are shown by this angiotensin 2 and why do you need to inhibit its action. So that will be clear from the pharmacological actions of the angiotensin 2 by looking at what effects it is showing in our body. Okay, so coming to the first actions on the cardiovascular system. Now this is very important. So you need to understand what is the role of angiotensin 2 in generation of the pathophysiology of different cardiovascular conditions. That will make it clear why we need to inhibit the action of this angiotensin 2. Okay. Now, the most prominent action of this angiotensin 2 is vasoconstriction. Okay. That is, it will decrease the lumen of the blood vessels, right? Okay, so diameter of your blood vessels is decreased. Okay, so that is known as, that particular effect is known as vasoconstriction. So what will be the consequence as a result of this vasoconstriction? Vasoconstriction is decrease in the lumen of your blood vessels, right? So if the blood vessel diameter gets decreased, what will happen to the pressure of the blood which is flowing through? Obviously it will increase, right? Because something fluid which is moving through the smaller diameter segment, obviously the pressure will be higher, right? So this angiotensin 2 ultimately contributes to the increase in the blood pressure. So let us see how it increases the blood pressure. Okay, so either this angiotensin 2 directly acts onto your blood vessels to decrease the diameter, or it may act indirectly by enhancing the ADR or NA. Now what is this ADR and NA? Yes, it is the adrenaline and the noradrenaline, right? So these are the pressure catecholamines, right? These are also hormones, these are also neurotransmitters released at the sympathetic system, right? So we are all aware that this particular neurotransmitters, or you can call it a hormone, it acts as both, right? So ADR or noradrenaline, these are catecholamines which are normally found in the body, normally released in the body, and they have pressure action, right? They can increase the blood pressure. So indirectly angiotensin 2 enhances the release of this adrenaline and noradrenaline both from adrenal medulla and from the adrenergic nerve endings. I told you that it acts as both the hormone as well as the neurotransmitter, right? So their release will be enhanced, and that's how it will result in vasoconstriction. So also another action of angiotensin 2 is by increasing the central sympathetic outflow, okay, sympathetic system, okay, from your CNS. So it is also getting stimulated. So obviously the central sympathetic supply also is towards your blood vessels, correct? So it will result in the vasoconstrictor action. So this is how indirectly also angiotensin 2 is contributing towards the rise in the BP. Okay, so this vasoconstriction, it involves the arterioles and venules. So arterioles, you are all aware that it is mainly regulating your blood pressure, right? So it is involved in mainly the regulation of the blood pressure. So arterioles and venules, these are getting constricted in whatever place it is present. Okay, everywhere throughout the body it will get constricted. So this results in rise in the BP. However, because of vasoconstriction, the fluid from your blood vessel, now supposing if this is your blood vessel, right, or blood capillary or your arterioles and venules, so these are getting constricted, so these will become narrow, right? So it will have a squeezing action on the blood which is flowing through. So as a result of this, the fluid will move out to the extravascular compartment. Okay, so now when the fluid moves out of your blood vessels, what will happen to your blood volume? Obviously the blood volume, that is getting reduced. So decrease in the blood volume means what? Decrease in the BP, right? So this rise in BP which is produced due to the vasoconstrictor action, actually it is an acute effect. That is, it is a short lasting effect. The rise in BP which is shown by the angiotensin 2. Okay. Further, another action is to increase the force of myocardial contraction. Okay. Now, why it is increasing the force of myocardial contraction? Your heart muscles, your cardiac muscles will contract more forcefully. So obviously the contraction of the heart will also increase. So that's because of the increase in the calcium influx. Okay, so we are all aware that calcium is responsible for the contractile property of the muscles, right? So because of the increasing influx of the calcium, more and more calcium will flow inside the cardiac muscles. More and more calcium will be released from the sarcoplasmic reticulum, and it will increase the force of contraction of the heart. Okay, now though we have seen that sympathetic activity is increased, it has stimulant action on the sympathetic system, right? It increases the sympathetic outflow, stimulates the release of the transmitters. Okay, so because of this action, it can increase the heart rate. Yeah, actually the heart rate should increase, right? That's what is the effect of sympathetic system. But usually reflex bradycardia predominates. It will increase the heart rate but transiently. Now when heart rate increases, you know that homeostatic effects will come into picture, and automatically through the vagal tone your parasympathetic system will get activated and heart rate will be brought back to normal, right? So usually it is a reflex bradycardia which predominates rather than the increase in the heart rate. Now some more effects on the cardiovascular system is on the cardiac output. Okay, so cardiac output, that is the amount of blood which is pumped by your heart per minute, that is reduced. Okay, now cardiac work, this is increased. Okay, so these two effects are due to the rise in the total peripheral resistance. That is, the blood vessels, what is happening to your blood vessels? These are getting constricted, right? So total peripheral resistance is what? It is a resistance to the flow of the blood through this blood vessel. So when the heart is pumping these blood into the blood vessels, if the diameter is smaller, obviously the peripheral resistance is high to the flow of this blood, right? So these effects, that is decrease in the cardiac output and increase in the cardiac work, is mainly due to this vasoconstrictor action which adds to the total peripheral resistance. So your heart has to work harder to pump the blood through these narrowed blood vessels. So now obviously when the diameter of the blood vessels is reduced, what will happen to the blood which is flowing through? That will also get reduced, right? Okay, so these are some more effects on the heart. Okay, now angiotensin 2, it does not activate the latent pacemakers. Okay, so it does not have any arrhythmogenic propensity. Okay, Okay, so if at all it is there, it is very little. Okay, it is not significantly precipitating arrhythmias. Now, all these actions, when they persist on a chronic basis, it will have deleterious effects on your heart, okay, as well as onto your blood vessels. Now, constant rise in the BP or constant increase in the work of your heart, obviously it will induce the hypertrophy, that is increase in the size of the myocardium will take place. Hyperplasia, that is increase in the number of cells, will take place. Even there will be increase in the intracellular matrix production as well in your myocardium and your vascular smooth muscles. Okay, so this will be due to direct cellular effects which may involve the expression of proto-oncogenes or transcription of several growth factors will take place. So this is responsible for increase in the size of the organ, okay, or the increase in the number of cells, or the size of the cells itself may increase. Matrix formation may also increase. So several remodeling processes will take place over here. Okay. Apart from that, indirectly volume overload and increased total peripheral resistance, which is caused by angiotensin 2. This will also contribute towards hypertrophy and remodeling, that is abnormal redistribution of the muscle mass in the heart as well as the blood vessels. Okay, so ultimately what will happen? Angiotensin, when it persistently keeps the blood pressure high, or if it persistently keeps the blood volume high, obviously it will result in changes in the size of the organ, right? We have seen under the renal system as well. If the load on the kidney increases, obviously it will start increasing in size, right? Okay, to cope up with the functions. Okay. Now this long-standing hypertension, it will result in further complications, like it will result in hypertrophy, as we have seen increased blood vessel and intimal thickness will take place. Also ventricular hypertrophy will also be produced. Apart from that, fibrosis, that is a scar formation or dilatation of the infarcted area because of hypertrophy of the non-infarcted ventricles, ventricular walls. This may also be seen after the myocardial infarction. Even in case of congestive heart failure, there may be cardiac cell death which may take place. Okay, that is the cardiac cells may even die, and fibrotic transformation, that is scar formation, will take place in the heart. So all these are the complications of long-standing high levels of angiotensin 2. So as we can see that all of these changes which are taking place, these are an important risk factor for the development of the cardiovascular complication, may it be morbidity or mortality as well. Okay, so what is the basic idea over here? What you can conclude from here, that angiotensin 2 levels remaining high for too long time is not good for the cardiovascular system. Okay, so this is the basis for the development of various drugs which can inhibit this renin-angiotensin system, so that the level of angiotensin 2 which is generated, it will be kept low, and all these complications which are seen over here, these can be minimized. Okay, so this is to do with the pharmacological actions on the heart, the cardiovascular system, heart as well as the blood vessels. Next action is on the smooth muscles. Now angiotensin 2, it can contract most of the visceral smooth muscles in vitro, but this particular effect is not significant in vivo, that is in the actual alive person. Then next action is on the adrenal cortex. Okay, so adrenal cortex, it's a part of what? Yes, it's a part of adrenal gland, right? Where are adrenal glands located? These are suprarenal, just above the kidney these are located, right? Now this adrenal cortex, it is the outer part of the adrenal gland, right? Inside part is known as the middle part is known as adrenal medulla. Now this adrenal cortex, it generates various hormones, right? So there are 3 layers, and out of that, zona glomerulosa, it is responsible for the synthesis of aldosterone hormone, right? So this angiotensin 2 as well as angiotensin 3, Both of these are trophic to zona glomerulus. That means what? They will increase in the size and it will result in enhanced synthesis as well as release of this hormone aldosterone. Okay, so aldosterone, what is the role? We have already covered under the renal system. That is aldosterone, it acts on the distal tubules, right? If you can recall, under the diuretic drugs, we have seen that there are certain transporters whose synthesis is enhanced by the aldosterone. There are mineralocorticoid receptors on which this aldosterone will bind and it will result in synthesis of certain transporters for sodium. So sodium reabsorption will take place and in turn it will get exchanged with potassium and hydrogen, right? So under the influence of angiotensin 2 and 3, aldosterone generation will increase. Obviously the transporter generation will increase. So as a result of this, reabsorption of sodium and excretion of the potassium and hydrogen will be increased, and this effect is shown at very low concentrations, even lower than that which is required to produce the vasoconstriction. Then next effect is on the kidney. Okay, so now we have seen that through aldosterone also it affects the kidneys, right? It will alter the reabsorption of certain ions and excretion of certain ions. So apart from the action through the aldosterone, angiotensin 2 directly promotes the sodium and hydrogen exchange in the proximal tubule. Okay, this is the one. As we have seen, there is a transporter, sodium hydrogen exchanger or antiporter. So this particular transporter's activity is enhanced. So what will happen? It will increase the reabsorption of sodium ions. Now along with that, chloride ions will be taken up and also the bicarbonate reabsorption will also promote it indirectly. So this we have already covered under the diuretic drug, so we shall not repeat all that again. Okay. Then next action is on the CNS. Okay. So the systemically administered angiotensin 2, it can gain access to certain areas of the brain, and by action on the brain it can induce the drinking behavior. That is, you'll feel more thirsty and you'll start drinking more of fluids. Okay, fluid intake will be increased. So also ADH release will be increased. What is ADH? ADH is anti-diuretic hormone. So we have seen the role of ADH. What it does? It promotes induces the insertion of aquaporine channels, and it will increase the reabsorption of water in the later parts, right? Anti-diuretic hormone, that is, it will decrease the output of urine, right? So that urine output is reduced. So what will happen as a result of both of these actions? Fluid load of the body will increase, right? So here by drinking more and more fluids, you are adding to the body fluids, and under the influence of ADH, more and more reabsorption of water is taking place. So ultimately what will happen? Plasma volume expansion will take place under the influence of both of these actions. Okay, so these are the actions of angiotensin 2 which are mediated through the CNS. Apart from that, it can also increase the central sympathetic outflow. Remember, central sympathetic outflow, under the actions we have seen that it also contributes towards the pressure response, right? Because ADR and NA which are released, they have pressure action, right? Vasoconstriction will be produced and it will result in increase in the blood pressure. Next action is on the peripheral sympathetic system. So we have seen so far that central sympathetic outflow is stimulated. Not only that, angiotensin 2 also increases the sympathetic activity in the periphery as well. So it can increase the release of ADR from the adrenal medulla. Also it can stimulate the autonomic ganglia. Also it can increase the output of these noradrenaline from the adrenergic nerve endings which are present in the periphery. So that's how it can enhance the peripheral sympathetic actions as well. Okay, so this is to do with the pharmacological actions of angiotensin 2. Coming to the angiotensin receptor. So let us see through which receptors exactly this angiotensin is mediating its action. So there are specific angiotensin receptors which are meant only for binding of this angiotensin, which are present on the surface of the target cells. So there are two subtypes of these angiotensin receptors which have been discovered, that is 81 and 82. So this has been discovered and differentiated. Losartan, it's a selective antagonist for 81, and this particular drug PD123177, this indicates that it is being developed. So this is a selective antagonist for 82 receptors. Now both of these receptors, whether 81 or 82, both of them these have been studied in detail, and these have been found to be G protein coupled receptors. So these operate through phospholipase C, IP3, DAG pathway. Okay, so all of these actions of angiotensin 2, these are mediated through 81 receptor. So this is important for us. Okay, angiotensin, one subtype of receptor needs to be inhibited if at all you want to block the actions of angiotensin 2. So this should be your target because it is selectively mediating all of its actions through this 81 subtype of the receptor. Then coming to the pathophysiological role, so let us see what exactly are the roles. So we have seen pharmacological actions on various systems. Now let us see what are the pathophysiological roles of angiotensin 2. Now first one is the mineralocorticoid secretion. Okay, so as we have seen that it acts on the adrenal cortex and angiotensin 2 and also angiotensin 3. It stimulates the secretion of aldosterone from the adrenal cortex. So it is even trophic to the glomerulosa cells. So it will increase the synthesis and release of the aldosterone secretion. Okay. Then next is a electrolyte, blood volume and the pressure homeostasis. So we, as in the introduction, we have seen that angiotensin 2, it is found to be involved in the electrolyte, blood volume and the blood pressure homeostasis. So let us see how exactly it plays a role in the homeostasis of all these three aspects. Now, electrolyte balance, it needs to be maintained, right? So certain electrolytes, ionic components of our body, it needs to be maintained within a normal range, right? Even the blood volume and the blood pressure as well. Otherwise it will result in the complications, right? If these are not maintained, certain pathological conditions may get precipitated. Okay, so this homeostasis maintenance is taken care of by this renin-angiotensin system. Now when this system will get activated? So whenever there is low blood volume or low blood pressure or decrease in the sodium content, as we have seen in the diagrammatic representation of the system, we have seen that lower volume or lower pressure or lowered sodium content of the body, it will induce the release of this enzyme renin. Okay, now how exactly this renin release is promoted? That takes place by three mechanisms. Three mechanisms are responsible for increase in the renin release. So let us discuss all of these one by one. First is the intrarenal baroreceptor pathway. Okay, now baroreceptor means what? The ones which are regulating the pressure changes, right? So whenever there is decrease in the pressure, obviously the tension onto the arterioles, the glomerular arterioles. Remember the glomerulus, it has one blood vessel which is entering into it, right? So that is known as efferent, and then further there is a capillary network which is present, right? And there is one capillary which is coming out of your glomerulus, right? So this is your Bowman's capsule, within which these glomerulus is present, that is a network of capillary. So one capillary is entering within this glomerulus, and that is known as the efferent arteriole. Okay, and it turns out to be into a network of capillary, and one arteriole which is coming out of this capillary network is the efferent arteriole, right? So this is the unique in that it is a capillary network present in between two arterioles, right? Usually the capillary network is present in between an arteriole and a venule, right? But over here, this glomerulus, which is a part of your cardiovascular system as well as your renal system. So this intrarenal baroreceptor pathway, it operates through increased production of the prostaglandins by decreasing the tension into these afferent glomerular arterioles. Okay, so it operates partly through increase in the production of the prostaglandins as well as via stress sensitive ion channel. So this is how it is operated. Next pathway is the macula densa pathway. The macula densa, as we have seen, that these are the modified type of cells which are present in the tubular wall, right? So macula densa along with the juxtaglomerular cells, both of these comprise of the juxtaglomerular apparatus, which is continuously monitoring the ionic composition of the fluid which is flowing through your kidney tubules or the renal tubules, right? So whenever there are low levels of sodium or chloride ions into the tubular fluid, it is sensed by these macula densa cells, as we have already discussed under the renal system. Okay, so this is how it will trigger the release of the renin. So by sensing the low levels of these ionic compositions, it will result in the synthesis of the PGs. Now, how it is occurring? It's by activating the enzyme COX-2. Okay, COX-2, as you are all aware, it is involved in the synthesis of the prostaglandins, right? So PGE2, PGI2 levels are enhanced. Okay, both by enhancing the COX-2 as well as neuronal nitric oxide synthase. Okay, now neuronal nitric oxide synthase. Now, what is the role of nitric oxide synthase? Obviously, to generate more and more nitric oxide. So this in turn will enhance the PG synthesis. Okay. So both of these, COX-2 as well as activation of the nitric oxide, will result in increased PG synthesis. Okay. So also PGs will be locally released and it will act on the juxtaglomerular cells to promote the release of the renin, right? Renin, we have seen that it is produced by the juxtaglomerular cells, right? So this is the second pathway which increases the renin release. The third pathway which is responsible for renin release is beta adrenoceptor pathway. So the baroreceptors and other reflexes which are operating within the kidneys, these will increase the sympathetic impulses to the juxtaglomerular cells, okay, activate the beta 1 adrenoceptors, and this is how it will enhance the release of the renin. That's by increasing the intracellular CAMP concentration. So these are the three basic pathways by which renin release can be increased. Now, when renin release will increase, what will happen? What is this enzyme responsible for? Generation of more and more angiotensin 2, right? So renin, which is released in your blood circulation, it converts angiotensin nogen to angiotensin 1, and angiotensin 1 is further converted to angiotensin 2. Now this will be responsible for acute rise in BP because it produces vasoconstriction, right? Apart from that, long-standing effects by direct as well as indirect increase in the sodium and water reabsorption will also be produced, right, as we have seen under the actions. So this rise in BP in turn, it inhibits the release of renin. Okay, so this is a feedback mechanism which is controlling the amount of renin which is getting released. Right? So far we have seen what is the mechanism which increases the renin release. Now let us see what are those mechanisms which are controlling the release of this renin. Now continuously if the renin levels are high, what will happen? More and more angiotensin 2 will be generated and it will result in the rise in BP and other deleterious effects, right? So we do not want this to happen, right? Our body will not keep quiet. Obviously, there will be a negative feedback mechanism which will decrease ultimately the release of the renin, right? Now, this particular system which is operating, it's a long loop negative feedback mechanism. Why? Because Renin, which is released by the kidney, it circulates through your bloodstream, generates your angiotensin 2, and this increased angiotensin 2 in turn will cause rise in BP and changes in your electrolytes, and this rise in BP in turn is responsible for decrease in the renin release. So it's a long process, right? That's why it is known as long loop negative feedback mechanism. Okay, whereas we have seen that there is also angiotensin which is generated within the kidney also, right? So it produces local regulatory effects within the kidney. Intrinsic local renin-angiotensin system will operate within the kidney itself as well, right? So this leads to the another negative feedback mechanism which operates within the kidney. Now here circulation was involved, right, in the first system which we have seen. But here within the kidney, whatever angiotensin 2 is produced locally, this activates the angiotensin 1 receptor in the juxtaglomerular cells, right? So this activation in turn will inhibit the renin release directly. So this is a short loop of negative feedback mechanism which is operating within the kidneys. Okay, so this ultimate system which is operating, it will help to keep your BP within a stabilized range. Okay, so this feedback mechanisms also help in prevention of continuous rise in BP which can be produced. Okay, so this regulation of renin release, it has a very important pharmacological implication. So as you can see that renin release, when it is increased, angiotensin 2 generation will be increased, so obviously BP will rise, and also we have seen other deleterious effects. So what you could do or how this could help in the pharmacological aspects, how you can utilize this particular information for pharmacological purpose, okay, to treat a particular condition, or what could result in the ultimate alterations of the system. Okay. So these are some of the implications. So, as you can see that if AC inhibitors or AT1 receptor antagonists, these are given, what will happen? It will enhance the renin release. Now why it will enhance the renin release? That's because both of these, it will interfere with the short loop and the long loop of the negative feedback mechanism, right? So this negative feedback mechanisms will get suppressed and ultimately it will result in increase in the renin release, right? Next is vasodilators or diuretics. These also can stimulate the renin release. Why? Because these two drugs, they lower the blood pressure. So when blood pressure is lowered, renin release will increase, right? Okay. So this is another implication. Then next is loop diuretics, okay, high ceiling diuretics. They can increase the renin production. How? That's because they reduce the entry of sodium and chloride into the macula densa cells, right? So loop diuretics, what they do? They promote the excretion of the sodium ions from our body. So levels of sodium will be low. Ultimately, the macula densa will sense that and it will start increasing the renin production. Next is central sympatholytics and beta blockers. So we have seen what is the mechanism of beta receptors in the renin release. So obviously, if central sympatholytics are given or beta blockers are given, obviously the renin release will be decreased because they will interfere with the beta adrenoceptor pathway. Okay. Then next is NSAIDs, okay, including the selective COX-2 inhibitors. So also NNOS inhibitors. What is this? Neuronal nitric oxide synthase. So if this enzyme is inhibited or COX-2 enzyme is inhibited by NSAIDs, what will happen to the renin release? We have seen that COX-2 results in the stimulation of COX-2 or stimulation of this nitric oxide synthase. NO will be generated and ultimately PG synthesis will be enhanced. This is how renin release is increased. So obviously NSAIDs, by inhibiting this enzyme or the inhibitors of neuronal nitric oxide synthase, it will inhibit the PG synthesis production and in turn it will decrease the release of the renin. Okay. Next pathophysiological role is development of the hypertension. So this renin-angiotensin system, as we have seen that it is involved in the renovascular hypertension development, right, through the plasma renin activity. This remains high, right, in most of the hypertensive patients. Okay. So in the patients who are suffering from essential hypertension, this particular plasma renin activity, it may be raised or it may be low, yet this system plays a very important role in maintenance of the hypertension, right? So ACE inhibitors, these can considerably lower the blood pressure in the hypertensives, right? Why? That's because AC is involved in the conversion of angiotensin 1 to angiotensin 2, right? So this enzyme, when it is inhibited, obviously it will lower the blood pressure. Okay. So also we have seen what is a correlation between circulating angiotensinogen level. If these levels are high, obviously the angiotensin production may also be increased. So this particular high levels of angiotensinogen in the blood circulation is also found to be correlated with the essential hypertension. Okay. So also there are several evidences for development of pregnancy induced hypertension, that is known as preeclampsia or toxemia of pregnancy. So here there are autoantibodies, okay, which are produced, which can activate this AT1 receptor. That is how the hypertension is produced in the pregnant ladies. Okay. So also we have seen what is the role of angiotensin 2 in the development of hypertrophy and remodeling of the heart and the blood vessels. So this could contribute to the development of the hypertension. Next pathophysiological role is the secondary hyperaldosteronism. Okay, so RAS system, as we have seen that it increases the aldosterone production and release, right? So it has trophic action on the zona glomerulosa or level of or this layer of the kidney, and that's how it can increase the aldosterone level. So hyperaldosteronism can be produced. Next is on the CNS. Okay. So angiotensin 2, it can be produced in the brain locally, and it may play a very important role as a transmitter or a modulator. Say for example in regulation of thirst or hormone release or sympathetic outflow, central sympathetic outflow. So all these actions may be mediated through the centrally synthesized angiotensin 2. Okay, now as we have seen the actions and the pathophysiological roles of angiotensin 2, these are not very good, right? So this angiotensin 2 actually cannot be utilized commercially. It is not used clinically for any condition because it rather is involved in the pathophysiological roles, right? So what could be the conclusions of inhibition of the renin-angiotensin system? Obviously, by inhibiting this particular system, the levels of angiotensin 2, these can be kept low and many cardiovascular complications can be minimized. So inhibition of the system plays a very important role. So this has led to a number of compounds which has been developed which affect the system at various levels. Say the first type of drug is the sympathetic blockers such as beta blockers or adrenergic neuron blockers or central sympatholytics. All of these will decrease the renin release. We have seen how these are involved in the release of renin, right? So renin release can be decreased. So obviously when renin is decreased, the conversion of angiotensinogen to angiotensin 1 is decreased, and obviously the formation of angiotensin 2 will be ultimately reduced. The next is the renin inhibitory peptidase or renin specific antibody. So these will block the actions of renin. So thereby the rate limiting step, that is generation of angiotensin 1 from angiotensinogen, will be blocked. So angiotensin 2 also will not be formed. Next type of inhibitor is angiotensin converting enzyme inhibitor. So this is the enzyme involved in the second step, right, wherein angiotensin 2 is generated from angiotensin 1. So when this enzyme is inhibited, obviously generation of this active principle will be inhibited. Next is angiotensin receptor antagonist or AT1 receptor specifically, because we have seen that actions are mediated through AT1 receptors, right? So ARBs, these are popularly known as, okay, angiotensin receptor blockers. Okay, so these block all the actions of your angiotensin 2 on the target cells which are mediated through this receptor. And the last is the aldosterone antagonist. Okay, so obviously the mineralocorticoid receptors will be blocked by this aldosterone antagonist. Okay, that is the spironolactone. Okay, neplerinone. These are the two examples. So these will block the mineralocorticoid receptors, and all the effects mediated through the aldosterone which is released by this RAS will also be blocked. So this is to do with the angiotensin, its generation, the pharmacological actions, receptors, pathophysiological roles, and what is the significance of inhibiting this particular system and how it can be inhibited. So this is the reference for my presentation. Thank you for watching.